Thank you for the invitation. I'll assume people can hear me unless somebody objects. Yeah, my same feelings. I wish I could be there, but thank goodness for Zoom, so we can still share some of our results. I'm going to talk about this, what I call the 2' hydroxyl barrier, which is a sort of challenge that's emerged for us and other groups trying to achieve sort of really diverse or full chemical modification of CRISPR RNAs and sort of our biochemical approaches that we've taken. And a little bit of the story is chronological, so sort of how things came about for us. Okay, if we can, there we go. Cas9 is a prototypical CRISPR effector enzyme, many of which, or all of you, should know about at this time. And thank you to Mark for a good introduction. Cas9 doesn't work by itself or alone. It actually works, partner in crime as a CRISPR RNA that guides substrate specificity. You can think of it as a sort of essential cofactor. And this guides substrate specificity to a double-stranded DNA, and the Cas9 will recognize this NGG protospacer adjacent motif or PAM sequence, and then directly adjacent to that, you need complementarity to the guide RNA. And if you achieve that, you get cleavage, double-stranded DNA cleavage. So the power of this really for biotechnology, making custom cell lines, and ultimately therapeutics is that you create these double-strand breaks that Mark told us a lot about, either through NHEJ or inserting specific unique new sequences through homology-directed repair. And the real beauty of Cas9 is it now is helping us to sort of approach one of the last major frontiers in medicine, and that's genetic diseases. So thousands of which are incurable. And so gene therapies have been around for a long time, but CRISPR is making this more efficient and more programmable. There's a lot of ways to introduce CRISPR. Let me actually turn on a pointer. So there we go. Lots of ways to introduce CRISPR. This is sort of a leading or popular method for therapeutics, and that is to package an mRNA that will encode for the Cas9 because it's a rather large protein, and then co-package or package separately also a guide RNA. So this would be a lipid nanoparticle sort of delivery approach to target specific tissues. Very exciting. Only a few years ago, we were doing sort of the first infusions in humans, and now it looks like very likely that we'll have some FDA approvals maybe as soon as this summer, such as for sickle cell disease, and there's multiple companies converging on FDA approval very soon. Lots of money, lots of time and effort's been poured into CRISPR, so it feels like it's going to succeed. There are still some challenges, long-term risks. We've made a lot of headway in how to predict off-target effects and sort of mitigate them, but they're still difficult to fully predict and eliminate. And that still is a challenge. It's a concern. If you're thinking therapeutically, you certainly don't want to introduce an edit in the wrong gene or the wrong tissue that can give the patient cancer years down the road. So these are not, they didn't evolve in humans for therapeutics. They evolved in bacteria, so they can cause immunogenic effects, and this can raise issues with long-term expression of the Cas enzyme or sort of repeated dosing. As a biochemist, I like to think about biochemical challenges and how we can better understand them, obviously to leverage for therapeutics or other applications. So some biochemical challenges that I think would be helpful to learn more about and understand is the efficiency of the enzyme. Some sequences are much more efficient at performing gene editing or even cleavage than others. Of course, we have algorithms and tools to help us figure out what these are, but also at the same time, classic nucleic acid therapeutic approaches are done where you just screen through the guides and sequences until you find the right one. It would be helpful to learn more about the underlying mechanisms chemically. Very importantly, of course, is off-target effects, which we talked about, and how to address those and predict those. And then, of course, stability, the half-life or the durability of the enzyme in vivo or even inside of cells is difficult to control. So what we've decided to do in my lab is we've got this sort of ongoing project where we're looking at these challenges from a chemical modification of the guide RNA perspective and thinking about efficiency, specificity, and stability. And we all know that chemical modifications of nucleic acids is a great way to sort of probe these systems and, find out what's tolerated or even beneficial. Lots of CRISPR systems you can explore. We've done some work with Cas12a. Today, I'll just talk about Cas9. And when approaching chemical modification of the guide RNA, you have the commonly used single guide RNA system, but that's actually an artificial structural modification of the naturally occurring dual guide RNA. So the dual guide RNA system that Cas9 from S Pyogenes uses has a CRISPR RNA and then a second tracer RNA. We've explored chemical modifications in tracer RNA. Today, I'm really going to focus on the CRISPR RNA because I think it helps it helps exemplify the challenges that we're facing, and I think it is smaller, easier to modify, and study with more techniques. So this is sort of the dual guide structure, and we're going to focus mostly on the CRISPR RNA. And our initial probe, when you want to start doing modifications, how do you begin? So smaller, simpler CRISPR RNA that we can study, and then we decided to start with DNA. As a probe. It's very affordable, lacks a 2' hydroxyl, so it can't hydrogen bonding with the protein or other molecules, and very flexible, but can assume the A-form structure. So it was a good probe. And what you see on the right is a panel that I just want to take a minute to go through, because this is how we present the results in a very visual way. So at the top is your sequence of the CRISPR RNA, and this is the color coding scheme that we're using for this particular panel. So the RNA bases are going to be orange and DNA is blue. And on the right-hand side is where we display sort of the activity. This experiment was looking at in vitro cleavage activity. So just the intrinsic biochemical activity of the enzyme, not gene editing. So we initially just made a series of modified guides, and we're looking to see where is DNA tolerated. So the control here is at the top, get about 50% cleavage activity with this particular guide sequence. If you convert the entire thing to DNA, you lose all your activity. Interestingly, though, when we started modifying it, we found that the loss of activity was due to over-modifying the guide region with DNA. But if you put DNA in this tracer RNA pairing region, also known as the repeat region, it actually was beneficial. We always consistently got higher activity than the unmodified. Also, RNA in the guide region is really only required in the seed region. That's where you initiate or nucleate assembly to the complex with Cas9. To your target DNA. And then, interestingly enough, too, we knew that there were these predicted 2' hydroxyl contacts between the CRISPR RNA and the peptide backbone of Cas9, and those are indicated with these asterisks at the bottom. If you take CRISPR RNA and you modify completely to DNA, but you retain those positions as RNA, you rescue some activity. So they are important. However, if you do the converse experiment where you keep the entire CRISPR RNA as RNA and you just change those positions to DNA, you don't lose any activity. So this suggests that contacts may be important, but they're not actually essential. And maybe it's the A-form structure or helical structure that the RNA nucleotides are inducing. So we also looked at some different sequences. We saw similar trends. And I want to point to this particular design or modification scheme we call J. And that was the one here where all of the tracer pairing region was DNA, but we kept the guide region as RNA, and that one always gave higher activity. And we saw the same trend with different sequences. So we decided to look at this a little closer and try and tease out what's going on. Just a little foreshadowing, we didn't fully solved it, but we've done some experiments, and this has been set aside temporarily, and now we're sort of interested again. So we'll get back to this. But the first thing we found was that it doesn't speed up the reaction. It actually just allows the reaction to go to greater completion. It just enables more cleavage. It doesn't impact the assembly of the complex. It looks like the complex still assembles just fine. In fact, an all DNA version, which is the control here called E2K, also can assemble a complex with no problem with Cas9. The problem arises for the control, all DNA control, when you're trying to actually bind and engage your target DNA substrate. So it can't do that if it's all DNA. But if you have DNA in that tracer pairing region, shown in orange, you really have little or no impact on substrate binding. And then also you can look at the stability of the complex by thermal stability. And interestingly enough, the all DNA E2K again assembles a complex just fine with a slightly reduced thermal stability. And this E2J version does the same thing. So a slight decrease in thermal stability. Unclear exactly what's going on, and we've left to conclude that this got to be affecting the dynamics or conformational state transitions in some nuanced ways, and we're following up on these designs. But what we did next was we actually wanted to look at more diverse chemistry. DNA was helpful, but it left us wondering if it was just needs some two prime hydroxyl hydrogen bond, some hydrogen bonding like 2' hydroxyl, or if it just needs A form helical structure. So are there modifications that can get around this and allow us to modify some remaining positions? So A form helical structure, what's the importance of these 2' OHs, and also flexibility, which we know DNA introduces. Do this, I teamed up with an old friend, Masad Dama, and Dan O'Reilly is shown here. He was a graduate student at the time. He's now a postdoc with Anastasia Korvova. They made a series of oligos with us that had a variety of modifications. And we wanted to look for analogs that could maintain an A form-like or RNA-like structure, some that were DNA-like, so 2' FANA. Many of you know maybe that Masad Dama, that's his favorite modification. He always wants to put FANA in oligos, but it's a great DNA mimic. It just is not flexible like DNA. And then some other more flexible modifications. But what we found was that a lot of modifications were very well tolerated. So again, on the right side, you'll see our same layout, plot of the data, but what you should notice is there's a lot more colors involved. So the RNA, here's our control, there's our standard activity. And there were a number of versions in this just series alone that had high activity, similar to the unmodified. And the key thing is these ones I've boxed or highlighted contain no RNA nucleotides, no native RNA nucleotides. So again, there's no intrinsic necessity for the enzyme to require RNA, at least in vitro. And just some other trends, RNA, of course, was much preferred. RNA-like analogs were much preferred in the guide region, DNA much preferred in this tracer pairing region. And then this central region, which happens to be where these 2' hydroxyl contacts are located, seem to be the most sensitive to modification. This was in vitro cleavage activity, and now we want to move to cell-based editing. So we looked at some that worked pretty well in vitro, and they give strong cleavage in vitro, but then when we tested them for cell-based editing, they were pretty dismal, little or no activity. We then made a simpler series where we just modified 2 of these predicted 2' hydroxyl contacts, nothing else. Some of these worked quite well in vitro, and those significantly reduced activity inside the cell. So now we're wondering, why on earth is this activity not correlating? And is there a way to correlate this? And are these 2' OH, these RNA residue positions, is it the 2' OH contacts? What is going on with why the enzyme doesn't like to use these inside of cells? A lot of directions we could go, but one was to further explore chemistry, and that was to look at modifications that could ideally maintain or preserve some of the 2' OH's ability to make hydrogen bonds, either to receive them, donate them, or both. So this included 2' amino and 2' arabinose, which just has the OH and the up stereochemistry. And then these interesting structures, ring structures that are called oxapane, and they have multiple, at least 2 positions where you can get hydroxy, you can get some hydrogen bonding. Same sort of approach. This time, what you're going to see on the right-hand side is in vitro cleavage in orange, and the blue bars are gene editing, and that's editing of an EGFP gene that we can measure quickly. And we focused our modifications exclusively on these 6 critical, we call them critical positions. And everything else outside of that, those 6 positions, is either going to be all RNA in red, here's our control, or all 2' fluoro in black. And I'm not going to go through all these, but some interesting ones were this one we call SJ1 that only put fluoro in those 6 positions, and we lost most of the editing. Not all of it, but most of it is gone just by changing those 6 positions. If you do the flip experiment where you put 2' fluoro everywhere and just RNA at those 6 positions, it works as good or better than the native CRISPR RNA for gene editing. Unfortunately, oxipane and 2' arabinose just were not very good. There was very low editing or no editing for most of those. Those may also be with some steric considerations. Look more closely at this. Is it a particular position? Is it all of them? Is it an additive effect? We decide to do just a simple walk of modifications through those 6 positions. And when we do that, we find that we see some positions are more well tolerated, position 4. Others are less tolerated, more sort of on the termini or outside of this stretch of positions. This was with 2' H, so DNA. LNA had a very similar trend, although for some reason slightly lower across the board. 2' fluoro was basically moderately tolerated at all positions, pretty good at the first position, but less at the 4th position. And 2' amino had a very similar trend to DNA and LNA, but just overall a little bit lower. The point is here, you may be able to combine certain chemistries to achieve high activity by understanding which positions tolerate what kind of chemistry. Think it's not just 2' OH. Some positions may require hydrogen bonding better. Others, it's more of a steric shape. And so but the problem is looking at crystal structures doesn't quite make this obvious. But we'll get back to structural roles here in a minute. But first, we want to test some sort of functional hypotheses. The first was whether or not it's the stability of the RMP complex. Are we assembling these complexes, but then they fall apart easily, or they just are not very stable? So we just selected a couple chemically modified versions here that had some different varieties of cell-based editing, but all were quite active in vitro. And we performed thermal denaturation or UV melts of these. And we can pick 2 examples here. This is SJ3, has excellent editing inside of cells, and it has this a little bit of an increase in its thermal stability over the CRISPR-EG control. Then we can look at SJ16, which has no editing and also has very stable complex. So, the verdict's not out, but it does not appear that there's a clear correlation with gene editing for thermal stability. Another hypothesis we had was whether it would be in vitro cleavage activity, if that could correlate. So maybe it can get in there, can bind to the target, but then its kinetics are slowed due to cellular environment or something to that effect. And so we would hope to see that with kinetics in vitro. Typically, our cleavage assays, we have an endpoint, maybe 15 minutes or an hour. But if you do a time course, you can get at least some very rudimentary idea of the kinetics or the timing of the cleavage. And in this case, we can pick 2, one that has good editing, one that has poor editing. And we find that there's no correlation. The one that edits poorly appears to have excellent cleavage in vitro, and it's quite fast. So unfortunately, same conclusion. Looks like the kinetics of the system may not really be the limiting factor. I will just add this is, of course, not true enzyme kinetics. And one of the challenges is that Cas9 is not a true multi-turnover enzyme in vitro. There are experiments that can be done, but we haven't teased out kinetics better that we haven't bothered with at the moment. But what looks the most promising is target binding affinity with editing efficiency. So again, we picked this series of oligos that have declining editing efficiency, and then we look at their ability to bind the target, and they have declining target binding affinity. We have some more modification schemes that we're testing. We really like this correlation. It makes sense because the substrate, the double-strand DNA inside of cells, is a bit different than the DNA that you have in the test tube. And so we think this is correlating. And I think that would be nice if you could do a simple in vitro predictor of activity that would correlate or translate to good editing efficiency. So I think there's something going on more structurally with the protein. We think it's some nuanced effects, but it also could be some large perturbations in structure or how the RNA is binding or how the DNA is interacting with the RNA once you've assembled the RNP. We picked SJ1. This is an example where the 2' fluoros at the 6 positions and pretty low editing activity compared to an all-native CRISPR RNA. And with the help of a collaborator, Sergei Korolev at St. Louis University, we solved a moderate resolution, 3.4 angstroms of this modified SJ1 structure. And here's just a map of the electron density. And we've superimposed into it a better map of a 3 angstrom structure that we've solved. These are dual guide structures of an unmodified version. We'll just point out there are no dual guide structures available. No one has solved a dual guide structure before. Turns out it looks very similar to a single guide structure, but there are some changes and there's some small domain changes and some other features that we'll publish pretty soon. The interesting thing that we found too was there are multiple conformations. Now, normally for a single guide complex, when you do try to do cryo-EM, you can get most of your particles are in a single closed conformation. Now, if you do this for a dual guide structure, about 50% are in the closed conformation and about 50% are in what we call sort of an open conformation that we haven't 3D reconstructed yet. But this SJ1 version, now we have a broader variety of particle conformations. There are a lot of artifacts that can occur, so we don't know what this means, but we do know that there's a variety of conformational states that we're seeing now. I'm going to just look real closely. We're going to zoom in at a part of the CRISPR RNA that has 2' fluoro that's base paired to the tracer RNA. And we can look at these 3 fluorinated uridines that are all side by side. And we do see some changes in the, the conformation of the nucleotides, but they're very small. So we're not sure if this is actually having major impacts. When we do the 2D classifications, like I had mentioned, there are a variety of different conformational states that are hard to 3D reconstruct. We have a great collaborator who helped us with some MD simulations in the past, and I think it's time to do some MD simulations to try and see if there's some energetics and conformational fluctuations going on here. But I think we've concluded with this modification study that there are some nuanced effects on RNP dynamics, maybe conformational effects. And we also don't think these are things that have been studied carefully, and so they're sort of unmonitored or uncharacterized conformational things going on that are important for enzyme activity. All right, I don't know what time I have left, but I think it's time to wrap it up. So I think if you could take home a couple messages, it would be that Cas9 is really not perfect. It does need to be tuned and engineered a little bit, especially for therapeutics. The guide RNA can be thought of as an essential cofactor, and it does play a role in controlling and regulating the enzyme's activity, and not in obvious ways. And I think that's borne out in the modifications that we've tried to do. Chemical modifications are great. They're classic for probing structure-function activity relationships, and hopefully we can understand this, we call the 2' hydroxyl barrier, or those key positions. We can understand that a little bit better, then I think that will help us approach full modification of CRISPR-Cas9. And I want to point out that Cas9 is one of a lot of different CRISPR systems being developed. We know that Cas12a has this very same problem where there's positions that can't be modified. Those are known to be those critical 2' hydroxyl contacts with the Cas12a protein. And there probably will be other RNA-guided systems developed, either that are natural or engineered, that people will want to use for therapeutics. So I think this problem is worth pursuing a little bit further. Thank you. Just want to thank a number of students and postdocs that have helped on these different projects over time, funding, and my collaborators, especially Masad Dama and Sergei Korolev. So I'll take some questions if people have any. Thank you.
Thank you very much, Keith. I have a question based on everything you've presented. I mean, there's clearly quite a bit of context dependence in terms of what is tolerated and what is not. What are your thoughts about what might be the most efficient way to identify the most optimal pattern of substitutions where you can be allowed to use 2'-fluoro and 2'-methyl and 2'-amino and so on, without having to make 100 million different variants and testing them individually.
Well, I don't know. I think I'm going to have you join our lab meeting so we can solve that. But well, I think we now know that the steric considerations are important, so shape and sterics, and I think we do need to look more carefully at the structure. So I think MD simulations are a cost-effective way to tease this out a little bit and give us really good candidates. There are chemistries that can be pursued a little further. There's a variety of, I mean, a large variety of diverse sort of exotic chemistries that people don't really typically use for nucleic acid therapeutics, but would be great for probes. And if one or two were found that did fit really well, then maybe those would be great for therapeutics. So we have a number of chemistries we're actually incorporating still. There's a variety of new ones. MD simulations are important, and we're taking a sort of also reverse engineering approach where we're going to use fully chemically modified guides, and now we're in vitro or sort of doing a sort of directed evolution of the Cas9 to see what amino acid changes are needed to allow it to accommodate and edit with these modified guides. I think that will allow us to look at maybe what parts of the protein are actually involved, and I suspect we're not going to see just changes that of amino acids that are nearby the guide RNA, but also other places. So those are some ideas.
Other questions for Keith? Any questions from the online attendees?
Yeah, this is Donald Burke. Keith, that was really nice. Quick question for you. You showed the structure or the model at least, comparing the 2' hydroxyl and 2' fluoro oligos. That one, yeah, exactly. When I see structures like that, I have to ask myself about molecular motion and dynamics. You know, these things look like they're pretty much the same thing, but yeah, you know, once they start moving around, they can get to be essentially the same. Have you tried doing this sort of modeling using strands that your data show you are failed architectures? Force it to have those modifications and then see whether they also fit into similar envelopes. Or is there anything about the modeling that would predict the failures as well as the successes?
Yeah, absolutely. So I could boil that question down to is okay. These things move around, and what I will say is the cryo-EM structure confirmed that there's no major gross structural changes. These are nuanced small effects, if that's it. But there's got to be something going on, right? And you're right. This could be just the conformer we've captured. This is not a high resolution crystal structure, even if it was. So I think MD simulations will help us, and that's exactly what we plan to do is model in DNA, fluoro, other things that we've tested, example, the ones that we've walked through, and then we can correlate functional data to the structural data, hopefully. That will give us some predictions, and then, of course, we'll build those and test them. Great point, Don. Thanks.
Yeah, thank you.